This is the report on the project AEROTOX, an EPPN network project between the Gregor Mendel Institute in Vienna and the Université Catholique de Louvain in Belgium. The aim of AEROTOX was to understand the genetic basis of autotoxicity tolerance in barley. As a background, barley is an allelopathic crop, which means that it produces and releases secondary metabolites that are able to inhibit the growth of neighboring plants. The risk that barley undergoes in this process is that it intoxicates itself with its own phytotoxin. We chose barley because it is a diploid species with a high-quality reference genome sequence. There are mutant resources available as well as natural collections, and as I just mentioned, it is an allelochemical donor. The two main allelochemicals in barley are Gramine and hordenine, besides a range of minor allelochemicals contained in root exudates. To be able to map the genetic basis of this trait, we used quantitative trait loci mapping, in which we want to identify the genetic determinants of this autotoxicity tolerance. For this, we use a population with trait variation and genetic diversity. This collection has been partially sequenced by our collaborators at UC Riverside in California. And the goal of this project was to gather the phenotypic data that could go along with this genomic data. Specifically, the population that we used is the so-called Composite Cross 2. This is a historical collection derived from the intercrossing of 28 parental landraces of barley that is maintained by the lab of Daniel Koenig at UC Riverside. These lines were intercrossed and then self-propagated in the field for almost 60 years in a natural evolution experiment. Prior to AEROTOX, we had gathered preliminary data on trait variability by growing lines from this collection and also the reference line in such a system as shown here, so growing barley plants on blue sheet paper in which the roots can be easily phenotyped because they grow along the paper and are nicely spread out. And we can supply chemicals to this hydroponic culture system. We did this, as shown here, for example, for Gramine in increasing doses, and we noticed that Gramine, which is produced by barley, is able to also inhibit root growth in barley, so there clearly is an autotoxicity happening here. And we wanted to establish the variability of this trait in the CC2 cross. For this, we germinated CC2 parental landraces in 5 millimolar Gramine and in control solution on this blue sheet paper system to measure root growth inhibition. And we noticed that there was a wide range of phenotypic diversity when it came to this trait as shown here. So here in order aligns with very low sensitivity to Gramine up to very high sensitivity of Gramine. This is shown in more detail here. For a selected subset of lines, for example, line number 5 had very high sensitivity to 5 millimolar of Gramine, whereas line number 6 shows almost complete insensitivity to this compound. Line 19 even shows an enhanced growth in Gramine conditions. In AEROTOX, we made use of the aeroponics system that is located at the Université Catholique de Louvain in Belgium, where plants grow on these suspensors and roots grow in air and can be treated with a spray solution containing our chemicals of interest. The project was divided into 2 steps. A pilot experiment in which a dose response analysis was done to determine Gramine sensitivity in a small scale setup with a concentration range between 0.02 and 5 millimolar of Gramine using the reference genotype Morax. And a large scale experiment with 2 treatments, one control treatment and one half maximal effect concentration and EC50 concentration of Gramine. In total, with 1,485 plants per treatment, spanning 145 genotypes, with the 28 parental landraces, a reference strain, and 116 crosses of the different parental landraces. This is what this looked like. So plants are grown on this platform, so they germinate in these containers here. And at the end of the experiment, so roots grow underneath, and at the end of the experiment, we can collect the tissue of these plants, which is manual labor, so we can separate the root from the aerial tissue, for example, to do subsequent metabolomic analyses of the tissue fluid. Currently, we are evaluating and analyzing the phenotypic data collected from this experiment. We are combining the phenotypic data with the genomic data that we have already generated to run association studies to ultimately map the genetic basis of autotoxicity responses. And finally, the data that was generated in AEROTOX can be combined with that of two other EPPN projects. One is PhenoHorde, which was completed in 2019 on the above ground phenotyping, and another one is RhizoTox, which was completed only a few months ago, where we grew the CC2 parental landraces in soil. Special thanks for this project go, of course, to Xavier Drey at the Université Catholique de Louvain, and also to Renaud Bourba, who ran the experimental work, particularly in the COVID situation where we could not be on site to run the experiments, and people in Louvain had to take over all the duties related to this project. Thanks also go to Zane Duxbury at the Gregor Mendel Institute in Vienna for conceiving parts of the project and also for analyzing a lot of the preliminary data. Thank you for your attention.